Welcome to the Proteomics video series, featuring interviews with 3 leading experts who'll share insights into the many facets of the proteomics field. Today, I'll be chatting with Professor Stephen Pennington, who is a full Professor of Proteomics at University College Dublin, about the use of proteomics in diagnostics and the workflows involved. Hi, Steve. Thank you so much for joining us today. So to start, could you briefly introduce yourself and describe your work at University College Dublin, particularly your involvement in proteomics and diagnostics?
Yeah, happy to do that. But firstly, thank you for giving me this great opportunity. So I'm going to go back in time a little bit, but move forward very quickly because it's been a fairly long career. I started out as a chemist, undergrad chemist and biochemist at Imperial College. And increasingly over the time, I realized how important that scientific grounding was for the work that I did subsequently. And then did a PhD in Cambridge in cell biology. And I'm really a cell biologist at heart. Got really interested in how proteins, provide for signaling molecules and control cell fate and behavior. And so that was really where I got passionate about proteins and the role of proteins in cells. So fast forward now to UCD, and the main thing I'm doing in UCD now is coordinating a public-private partnership from the Innovative Medicines, or now Innovative Healthcare Initiative, which is a sort of a large consortium focused on psoriatic disease, and can explain more about that a bit later. 27 partners, a large budget, and a 5-year, now going to be 6-year project. So that's what I'm currently. En route to that, then for a few times I've been president of the British Society for Proteome Research. For a while I was president of the Human Proteome Organisation, and I suppose one of the most exciting things for me in my sort of career was to have the opportunity to organise the HUPO, the Human Proteome Organisation annual meeting in Dublin in 2017. And before that began, we had a gala, what we called a gala dinner. And the after-dinner speaker at that gala dinner was Joe Biden. So that was quite and that was part of our attempt to try to, I suppose, spread the word about the excitement around proteomics.
So your work sits very close to the clinic. From your perspective, how is proteomics changing the way we think about diagnostics and disease stratification?
Yeah, so within Hippocrates, we are very close to the clinic. So a number of the centres, there are 27 partners in that organisation, a number of them are academic hospital-based centres, with clinicians, in this case rheumatologists and some dermatologists. And that connection, I think, is really important. And we may talk about that later. From my perspective, how we're thinking about diagnostics and disease stratification, I think increasingly we've now moved from where diagnostic tests measure a single protein, or maybe just 1 or 2 or 3 proteins, to actually measuring more proteins simultaneously to get a better sort of resolution of that stratification and maybe even ultimately personalization. So I think that the realization that we can measure multiple proteins simultaneously and incorporate those into diagnostic tests, I think is important. And then also I think combining that proteomics data with other data, including obviously genomics data, and digital biomarker data.
And to understand that impact, it helps to look at how proteomics fits into real-world clinical workflows. Could you walk us through what a typical clinical proteomics workflow looks like, so from patient sample collection through to data analysis and clinical interpretation?
Yeah, so I think there are 2 answers to this question. The first is that in a sort of a clinical setting, it's highly automated and has relatively high turnaround throughput and low turnaround times. So a sample, gets drawn by a phlebotomist in a phlebotomy department. It goes to the clinical diagnostics lab where it's in a barcoded tube, and it goes onto a fully automated track where it runs around a very large laboratory and then gets posted into the machines that can measure the proteins that have been asked to be measured by the clinician. The standard tests that are in current use. So that's one clinical proteomics scenario. But from a proteomics researcher's perspective, again, it begins with a sample. We're focused maybe on blood samples at this stage, but it could be a tissue sample. So if we're looking at liquid biopsies, a blood sample that gets generated, from which then serum or plasma is generated. and we begin the proteomics biomarker discovery pipeline. So those samples often digested with enzymes. Trypsin is the most common, and the peptides put into a mass spectrometer to try to measure differences in proteins between the samples across individuals. And those then become proteins of interest if there are changes in them. And in our workflow, we then develop targeted assays to those proteins of interest so that we can specifically measure multiple proteins of interest in a targeted assay, again, on a mass spectrometer. And then we use those measurements to evaluate whether, using other samples, whether the proteins of interest really do stack up in terms of addressing the unmet need. So that's one very sort of focused. It's focused on blood and it's focused on mass spectrometry. And that track that I described before, the automated track, at the moment, that's dependent on antibody or affinity-based approaches, so ELISAs. But moving to the future, I think there's an expectation, at least amongst some of us, that those tests that are done in hospital labs will be done on mass spectrometers. Protein tests will be done on mass spectrometers. And so we can measure many, many more proteins simultaneously.
Lovely, thank you. And I wonder, from your perspective, what are the most critical requirements for proteomics workflows to be viable in a diagnostic or clinical context?
I think those tests that would run in a hospital lab, obviously they need to be high throughput, they need to be really robust and automated. And I think automation is a key thing. And a number of mass spec companies have attempted to develop those kinds of systems and not succeeded, and spent a lot of money doing that. I suppose we're all hoping that the latest player in this game is Roche Diagnostics, and they develop these clinical analyzers, antibody-based clinical analyzers, and they're backing mass spec as being the future of these clinical analyzers. So that's essential. And if you move to the other end of the realm, again, I think the key things are being able to do the discovery experiments so that you can rely on the data that they generate. And that begins with the integrity of the sample and the numbers of samples that you're using and the approach that you're using both for the mass spectrometry and analyzing the mass spec data.
Lovely. And this sort of research involves close collaboration across different disciplines. What role do collaborations between clinicians, proteomics experts, and data scientists play in successfully translating proteomics into diagnostic use?
Yeah, so I think it's a really good question, and we could spend the rest of the day talking about it. Perhaps I think the key thing for me is that there's no point beginning the journey until you've identified a diagnostic test that you think is going to be of benefit to patients. So we call that start at the end. So identify that diagnostic test, and in our case, it's in psoriatic disease. So that involves interacting with rheumatologists and dermatologists. So you define the population of clinicians that you need to interact with, and for them to guide you, tell you what is most needed for their patients. So, areas of opportunity, and become very focused on that. Because if you just say, we've got a load of interesting samples and they came from patients treated with this or patients treated with that or this, and you haven't a clear direction at the outset, you're not going to get to the end point of a diagnostic test, I would argue. So those interactions are really, really important. And I suppose that, for me, was one of the exciting opportunities with Hippocrates, this consortium that we have, that it does involve close interactions with clinicians, with proteomics researchers, but also with genomics researchers and lipidomics and metabolomics, so, multiple omics, and data analysts. So part of the consortium is based in the Swiss Institute of Bioinformatics. So, and data analysts in some of our centers who are really experts in data analysis and the statistical methods that you need for combining the data if you're going to get to a test that involves proteins and genes and lipids and metabolites. So they're absolutely essential. I think one important, group of individuals that are part of this sort of network driving things forward are the patients themselves. So within our consortium, there are probably about 160 individuals, and 10% of those approximately are patients. And they can really, put a reality check in place, and also then, I suppose, guide you on what is going to ultimately be of most benefit to patients.
Amazing. Sounds great. Another question I had was, what do you see as the main barriers preventing wider adoption of proteomics-based diagnostics, and how might those be overcome?
Yes, and again, that's a really great question, and it's massive, and it obviously doesn't be based on my perspectives, but I think wider adoption means that the tests are being used. And so that's not just what we've spoken about so far as developing a test that's reliable and robust, but wider adoption requires another step, which is, getting it used in the marketplace, whatever the marketplace is, whether that's a hospital lab or a healthcare system or a niche CAP clear lab in the US, getting it incorporated into those and then disseminated to patients so that the tests get used, I think is really challenging. And I'm not sure I know the answer to that. I think it's really, it's customized to the test and the end users and the disease that you're tackling, because they all have different sort of characteristics. If you're developing protein tests for heart attacks, then the test needs to be super quick and almost at the point of the patient. Whereas if you're developing tests in inflammatory arthritis or complex autoimmune diseases or even cancer, the tests could be more sophisticated and take more time because the data's not going to be used the same day or the same hour. It'll be used in a consultation between the patient and their clinician, maybe a few weeks later. So that places different, emphasis on how you get those tests adopted. And I'm not sure that we're particularly good at doing that, getting wider adoption of these new tests, because there isn't a sort of a simple linear path to how you might do that. And it varies, it's almost as personalized as the the individuals that you're trying to provide the data from the test for.
So my last question for you is, looking ahead, where do you see proteomics having the greatest impact in clinical diagnostics? And what advice would you give to researchers aiming to move their work closer to the clinic?
So I think the future, if you ignore some of the challenges that we've spoken about, I think the future is really exciting and promising. And it would be, from my perspective, it would be multiplex protein tests that are robust and reliable in the liquid biopsy setting. I also think in terms of proteomics technologies looking at cells and tissues, there's a huge opportunity now with the development of, single cell and spatial proteomics technologies to identifying within tissues not just biomarkers that are indicative of disease, but also then, proteins that could be novel targets for the treatment of the disease. I think that's really, really interesting. And I suppose that segues a little bit, what advice would I give to proteomics researchers? My advice would be that they need to think really carefully about what it is they want to do, sort of to start at the end, decide what it is that they would like to have got out of the period of time that they're going to invest in that. I think and this is sort of university-based proteomics researchers, I think they need to get out more and not just in their own comfortable and out of their comfort zone a bit, not just going to their own proteomics meetings and meeting their old friends and all the rest of it. Those proteomics focused meetings, very few clinicians attend them for very obvious reasons. And I suppose also then try to focus. So don't try to do, and I think this is a mistake many of us have made in the past, is just spread ourselves too thin. Really to focus. And that means being really confident about, the bet that you're taking in terms of, the direction you're going and what to do. And then I've tried to do it, sometimes successfully, other times not, is just to reevaluate every 5 years or 10 years, depending on, how long things are taking, just to reevaluate where you are. And if things aren't working out, be prepared to start again, not just continue to on a track that's the evidence is pointing that it's not working.
Awesome. Great advice there. Thank you. That's all we have time for today, but thank you so much for joining us, Steve, for a really interesting discussion.
Lovely. Thank you very much.
Thank you. Thank you for watching this episode of the Proteomics video series. If you haven't already, be sure to check out the other 2 videos in the series, where we explore additional perspectives and topics from leaders in the proteomics field, including building reproducible pipelines of proteomics data and exploring what happens behind the scenes at an MS proteomics facility. See you next time.